Hello everybody, my name is Peter Keller. I'm the CEO of BlueWhale Bio. Welcome to our presentation. Advances. So let me introduce BlueWhale Bio to you. So we are a company founded by the University of Pennsylvania, out of the labs of Carl June and Jim Riley. And we focus uniquely on making CAR-T or immune cell therapy manufacturing more efficient. Our entire focus today is on activation technology. So we have developed a platform called Synecta Cell-Derived Nanoparticles, which is an activation technology that mimics the physiological stimulation that you would see with dendritic cells. We will explain later why this is advantageous. Our particles, Synecta T1 Cell-Derived Nanoparticles, are available commercially now for sale as RUO material. And importantly, we also started a clinical trial where we use Synecta T1 activation technology to stimulate CAR T cells and dose so far 4 patients in this clinical trial. So why is a company like us focusing on activation of immune cells? The reason is that this is a pivotal step in the manufacturing process that really defines what happens afterwards. If you do activation correctly, you will simplify your process, and I'm going to show you data that will also make it more robust and even more effective. Also important is that if you have the right activation technology, you can work with more starting materials. So you cannot only use isolated T cells and whole blood, you can also use exhausted T cells and will still see reasonable manufacturing yields with the right activation. Take a step back and look what's out there today. So today for activation, people use synthetic particles. Why particles? Because an immune cell needs to see at least 2 signals, stimulatory signal 1 and 2, to become activated. And this is today done by full-length antibodies for anti-CD3 and anti-CD28. And then basically there are 2 technologies. One is you take a polystyrene bead that has a magnetic coating, and covalently bound to it are the 2 antibodies to deliver the 2 stimulatory signals. But as these polystyrene beads are toxic, you also need technology to isolate them. That's why they are magnetic, so you can isolate the beads from the drug product. The other technology is basically a similar idea, but instead of polystyrene beads, you use biodegradable polymers. And this technology has also anti-CD3, anti-CD28 full-length antibodies, shorter shelf life, and it requires, to our experience, a bit more process development to get it to work. But what's common about those 2 technologies is it's synthetic material, rigid spheres, trying to concomitantly express or transmit those 2 signals. What is a requirement in the industry today is that the demands of the customers and developers for activation technologies will grow. And that's because we want to shorten the manufacturing cycle, less expansion time, so you need better activation for that. We have solid tumors which require a higher dose. There are more engineered cells, and for example, electroporation might exhaust cells. In autoimmune diseases, there's a risk for overstimulation or overactivation because they are relatively healthy cells in the autoimmune disease patients compared to a cancer patient. So that's why we think it's time to innovate the activation technology. And here we come with a completely new concept where we start from biological material and really try to simulate what a dendritic cell would do in vivo. However, we do it in ex vivo cell therapy manufacturing. So the starting point is a cell line, and that cell line has been carefully chosen for properties that are similar to the properties of a dendritic cell. Specifically, our cell line has adhesion molecules that really almost grab the T cell receptor or the immune receptor and create a tight bond between our technology and the immune cell. So once we have chosen the right cell line, we want to express the stimulatory signals. And we can now in the cell line express transmembrane molecules, which are ligands and not full-length antibodies. And that has advantages, and I will come to that. So we can not only put anti-CD3 and CD28 on it, we can also put more signals, including cytokines on our particles. And now we cannot use live cells in the manufacturing of CAR T cells or other immune cell therapies, so we need to disrupt the cells and isolate just the membrane with the signals and ideally nothing else. And we have a proprietary mechanical disruption process that achieves that. So I always compare it to like a diver goes under the water under high pressure, lung full of oxygen, goes up too quickly, the oxygen expands, and that's of course then unfortunately disruptive in that case. In our case, it's a good thing because we can basically preserve the membrane material with the desired immune signals, and that technically becomes a cell-derived nanoparticle. So our first product looks like that, in the cartoon at least. So we have an antibody fragment for CD3, OKT3 fragment. We have CD86 ligand instead of anti-CD28. We have 4-1BBL. So those are the stimulatory and co-stimulatory signals. And then we also express cytokines. So we have complex IL-15, IL-15 receptor alpha and IL-15 on the particle, and IL-7. So that's our founders say that's kind of the optimal activation machine. And so together with the adhesion molecules I was talking about, we can, with this composition, form what's called the immunological synapse. And that's just a more effective and efficient way to activate T cells, and they will grow much more readily. And that's basically the data and value proposition BlueWhale Bio has worked on. So number one, we observed we can radically simplify the manufacturing process. So where if you see here the standard process, you need to have your immune cells, you get your activation technology, the synthetic beads, you need to add cytokines, media, then you have to wait a day until the cells have become activated to add your lentivirus, then you need to refresh your cytokines every other day. With our technology, because we have the immunological synapse, we see activation much earlier. So therefore, we can also use the lentivirus earlier because only activated cells will take up the virus. So in our process, we propose the immune cells, Synecta T1, media and lentivirus on day 0, and then we don't do anything anymore until we can harvest. So we don't need to add cytokines in the way. And because the cells have been better activated, they will also grow faster. So we are typically, and that's not only us, also our customers observe, we are 3 to 4 days ahead in the growth curve compared to the standard process. So that will allow to shrink the manufacturing time, and that has, of course, cost advantages. So I will present to you later some of our results when we looked at direct cost savings with our technology. But it's not only simpler, it makes the process also faster, as I mentioned, but also more reliable. And I will show you data with exhausted T cells, and makes the quality of the product better because we avoid over-activation because we're using the ligands instead of the antibodies. And also for us, it was important that we tested not only in healthy donor T cells or immune cells, we tested in patient donors, and now also in the clinical trial. So we can really show those advantages with different material and different settings. So here, just as an example from healthy donors, we just look at the fold expansion as kind of the easiest measure for activation. And so we look at day 3, which is one of the shortest manufacturing cycles you can use. So we have Synecta in blue, and then the comparators in black and yellow. And you can see on day 3, we get already expansion. We have a 2-fold expansion on average here, whereas the other technologies don't get expansion. It's actually more of a retraction of the cell growth. And on day 9, this advantage continues, where we have significantly higher fold expansion with the Synecta technology compared to our competitors. And importantly to mention, to be fair, we don't add cytokines, soluble cytokines. For our comparisons, we always added cytokines to give them the chance to expand. So this is also an important slide because I told you we can use more starting material, we can have more robust and reliable manufacturing. So here from our founders at the University of Pennsylvania, we got patient samples from manufacturing failures. So these patients were enrolled in a CD19 CAR-T trial, but could not get those when they used the standard manufacturing process. So we got hold of those samples and in vitro, in a smaller scale, tried to expand those cells again with the existing technology or with, here in blue, the Synecta technology. And you can see only, as expected, the standard process didn't work, which we knew from the clinical trial, but with Synecta, the cells grew. So the investigator saw the data and said, I could have dosed this patient. So having the right activation technology can really make a difference between treatment or no treatment, and in case of advanced cancer, between life and death. So that was, of course, a reason why we want to do a clinical trial as fast as possible and show that this is applicable not only in a small scale, in a lab setting, but also in a clinical setting. So we filed a drug master file for Synecta T1 that basically describes our manufacturing process and our rationale for using this activation technology. Next thing was that the University of Pennsylvania had an open IND for an armored CAR-T. So this is a CD19 IL-18 construct, which uses a 3-day manufacturing process in refractory lymphoma and refractory leukemia patients. In the dose escalation phase of this trial, the university dosed 20 patients with the existing activation technology and saw that there were manufacturing feasibility issues with those 20 patients, so they had quite a few manufacturing failures, including the ones I showed before. So now the university decided to start a new cohort with the Synecta T1 as activation technology, referencing our drug master file. And they could do this without having a comparability study or opening a new IND. So it's important to note that in at least early clinical trials, we can really exchange activation technologies. And so far, we treated 4 patients in this trial with a 3-day manufacturing process and day 0 transduction. So we see already with 4 patients that we have significantly higher expansion than the previous 20 patients, and so no unexpected side effects. And we'll present more of the outcome data in a later presentation. So I mentioned also not only the advantages of robustness, speed, and quality of the cells, but also cost reductions. So we partnered with a company in the UK called Biopharma Services. They have a cost model called BioSolve, where they basically dissect the manufacturing process for immune cells or CAR-Ts and really know about all the materials that go in the process, but also know about labor for each and every step. And so we simulated with them a process where we use the comparator technology in a 12-day expansion and compare that with a 7-day expansion using the Synecta T1 technology. So to get started, we need to kind of make assumptions of what kind of factory we are looking at. So we simulated 25 parallel modules for a site that produces 300 doses a year. So typically, and according to their model, the total manufacturing costs to manufacture the CAR-Ts are around $120,000. And now we could ask the questions, how much would it cost in a Synecta process to produce these CAR-Ts? And so the answer was we can save around $23,000 in this process, driven by reduction in capital cost, reduction in labor cost, and material costs. So to give you some flavor of how we came to those savings, of course, if we can shorten the manufacturing cycle, because we see faster growth of the cells, we can produce more doses in the installed capacity, so that was a $6,000 saving. We also saw, I showed you the data with the failed manufacturing batches. We could reduce those manufacturing failures, we assume, just by 50% for the sake of the exercise, another $5,000 savings. And then, of course, we have shorter expansion, we have less manufacturing steps, that saves labor costs, saves material costs. So we have this model now very granular to demonstrate how a customer can benefit from this technology, not only quality-wise, but also cost-wise. And it is a platform because it's relatively flexible to express different immune signals on the outside of our particles. And so we start with Synecta T1 with all the immune signals we mentioned. The next product is Synecta T2, where we just use the OKT3 antibody fragment for the CD3 interaction and CD86, so it's kind of the minimal version, but has advantages for some customers who prefer, for example, to use IL-2. And then also very interesting, we have a particle that only has the membrane-bound cytokines, which has some interesting applications and also advantages over soluble cytokines. And one last thing to mention here for the pipeline is that because it's relatively easy to make those cell lines, we are also open to have modular, custom-made products, and we have some customers already talking and discussing with us how we can adopt the technology for their specific activation and expansion needs. So in summary, first of all, the product is available for sale on our website through the e-commerce solution, BlueWhale.bio. Don't type BlueWhale.com because that happened and you cannot buy it. So you find the offering there. And in summary, we can streamline the CAR-T cell manufacturing process. We can enable day 0 transduction, don't need any added soluble cytokines, have biodegradable material, so we don't need to get rid of the CDMPs at the end of the manufacturing process. It gives you reliable expansion, even in hard to expand T cells. and we get earlier cell division and overall a simplified process that we are now testing in a clinical trial. So thank you very much for your attention. I think we're just in time. Thank you.
Thank you, BlueWhale Bio.